So once we identified that kappa opioid receptors specifically within the nucleus accumbens shell mediate selective aggression that is key to pair bond maintenance, we next wanted to look at possible interactions between kappa opioid receptors that mediate aversion and the dopamine system that has also been identified as important for pair bond maintenance and motivated behavior. We also wanted to do this because these systems directly interact. Activation of D1-like receptors that mediate pair bond maintenance increases dynorphin, which is the endogenous ligand of kappa opioid receptors. So we thought it might be possible that interactions between these systems would be important for mediating selective aggression and therefore pair bond maintenance. And when we did these studies, we found that when we activated D1 receptors that would normally induce aggressive behavior but blocked the kappa opioid receptor system, we still got a decrease in selective aggression. So these studies determined that interactions between these systems are important for pair bond maintenance and probably are acting to mediate aversive motivation in general. So our behavioral pharmacology studies indicated that interactions between the D1 and kappa opioid receptor system were important for pair bond maintenance. So we next wanted to look at possible changes in dopamine transmission between sexually naive prey voles that are generally affiliative and pair bonded prey voles that are selectively aggressive towards novel conspecifics. And to do this study, we used fast-scan cyclic voltammetry, which is a technique that has been developed to measure real-time changes in dopamine release. So when we did these studies, we found that a depolarizing stimulation resulted in greater increases in dopamine concentrations in pair-bonded prey voles as compared to sexually naive prey voles within the nucleus accumbens shell. And this is important because this enhancement of dopamine release may be facilitating the activation of D1-like receptors that are important for selective aggression. So the next thing we wanted to do is to look at a possible relationship between dopamine release within the nucleus accumbens shell and selective aggression. So to do this, we ran, in a separate group of animals, we ran resident intruder testing prior to measuring dopamine release in our striatal slice preparation. And we did this in both sexually naive and pair-bonded prey voles. And we did this, we found a positive correlation between the amount of dopamine release and aggressive behavior. And this suggests that the more dopamine that's being released, the more aggressive this animal is. And that's indicating that dopamine transmission within the nucleus accumbens shell is playing a direct role in mediating selective aggression in pair-bonded prey voles and therefore pair bond maintenance.